Greetings, everyone. Earthlings. So I'm an exemplar. This is what you're supposed to use when you ask questions moving forward. And I've always wanted to hold it. So today, thank you all for coming. I'm happy to introduce Malin Pinsky. He is visiting us from Rutgers, where he is a newly minted associate professor in the Department of Ecology and Evolution and Natural Resources. Malin did his PhD with Stephen Palumbi at Stanford, graduated in 2011, and then he went on to do a postdoc as a Smith Fellow with Simon Levin and Jorge Sarmiento, right? Nice. So I want to invite Malin because he's a really creative and innovative thinker in our field. He's doing a lot of really cool work, especially lately. He's been cranking out some really interesting high-profile papers, and he's sort of interacting. His work started interacting with evolution, but a lot of ecology. And then what I really like about his work, he's always thinking about how this stuff can be applied in conservation. And so he's kind of bridging a lot of these disciplines. And I think he's sort of an up-and-coming leader in our field. And with that, I'm going to let him tell us all about life in a giant water bath, consequences for ecological dynamics in the ocean.
Cool. Thanks so much, Jake. I've had a fantastic day so far, hearing about the research that's going on, talking to students and postdocs and faculty. It's been really fun. This is my first time to University of Michigan, though I have been to Michigan before. In 2006, I drove from Maine and moved out to Portland, Oregon, so I came through Sault Ste. Marie. And actually, one of my first research experiences was actually on Isle Royale studying amphibians and population dynamics when I was an undergrad. So yeah, it's fun to be back in Michigan. So the world is filled with an incredible diversity of species, from microbes to plants, insects to birds and squid. Yet all of those species have to cope with strong variation in their environment through time and through space, whether that's temperature or light, nutrients, moisture. Some of the interesting questions include how, when, where, or even whether populations can persist in the face of that variation. We also know this is a time of rapid change in the environment as well, whether that's average temperatures globally, as I'm showing you here in this graph, or are there many other aspects of the environment that are changing rapidly, in part as a result of human impacts. And so I think some of the challenges for ecologists and evolutionary biologists is to integrate together the relevant processes so that we can then predict what do these changes mean for species and for communities. So species have a wide range of mechanisms for coping with environmental variation. A certain amount of variation in their environment is just tolerable from a physiological point of view. Behavior helps them avoid stressful conditions over smaller spatial and temporal scales. Over slightly longer periods of time, acclimation allows species to adjust their physiological limits. And then over generations, evolution can also rescue populations that otherwise might be extirpated by changing environmental conditions. If conditions in a single location are not suitable, populations can also colonize new locations, more suitable environmental conditions. Obviously, if all of these mechanisms fail, then extinction ends up being the result. We're not talking, though, just about single species, but actually many species all responding to environmental change at the same time. And so those interactions between species can also feed back to affect the action of any of these other mechanisms. And so those interactions between species end up becoming important. So we can think about these mechanisms as spread across scales of time and space, as well as biological organization, from individuals to populations and communities. And if we're interested in patterns like shifts in species distribution or persistence of species and extinctions, those are emergent patterns, right, from this complex system where individuals and species are interacting at fine temporal and spatial scales. And one of the consistent lessons from complex system science is that actually those emergent patterns are often not simple predictions from just an understanding of the fine scale dynamics, right? We have to understand how these processes operate across scales. So not only do we have to work up from the fine scale mechanisms, but also down from the emergent patterns until they meet in the middle. So one of the earlier efforts to put some of these pieces together was motivated by this question: how easily can animals cross mountain passes? And Dan Jansen's answer had a lot to do with this figure here, where we've got seasonality on the vertical axis, so the standard deviation of monthly temperatures across latitudes, so southern hemisphere, equator, northern hemisphere. High seasonality towards the poles, low seasonality in the tropics. And what Jansen pointed out, or at least conjectured, was that I'm supposed to move away from speakers, there's not feedback, I guess. In the tropics, where there's less seasonal variation, species have narrower ranges of temperatures that they can tolerate, and therefore it's relatively harder for them to move up and over cold mountain passes. In effect, mountain passes are higher in the tropics. So it's also been turned around by folks like Josh Tewksbury and colleagues to argue that tropical ectotherms are also most sensitive to climate change and warming, again because they have narrow ranges of temperatures that they can tolerate. But notice that this is, and that claim has sparked a lot of debate in the field, as well as a lot of really interesting research. But notice that this is really a debate about ecosystems across latitudes on land. So let's dip our heads under the surface of the ocean. This is where you could say that where 90% of the habitable space on Earth is found. And it's also an environment, though, with a very different evolutionary history, geographic patterns, physical environment as well, even histories of human impacts. And so I think this contrast between ecosystems, between realms, sets up really interesting opportunities to test ecological and evolutionary theory, and also to understand how the impacts of climate change vary across species and across ecosystems. So one of the things I want to do in my talk is explore some of these differences and what they mean for the impacts of climate variability and climate change and the ways in which species and communities respond. So on one level, in the ocean, we're talking about very deep diversity, so 33 animal phyla compared to 12 on land. The fluid environment is also very different. So specific heat of water, 4 times higher than for air. So it takes 4 times the energy to heat up a gram of water, a degree Celsius, than a gram of air. The oceans are also very big, 1.3 sextillion liters. So there's a lot of thermal inertia in the oceans. If we look at seasonality, I showed you the green line as well, so for land surface, so again, across latitudes. The blue line now is the ocean surface. So in the ocean, yes, there's low seasonality at the tropics, rising towards higher latitudes, but actually it drops towards the pole. So it's a qualitatively different latitudinal pattern in the ocean than on land. It's also, on average, 6 times less variability through time at the seasonal scale in the ocean than on land. There's also less variation across space as well. So relatively easy on land for organisms, mobile organisms, to move into the shade or into the sun and experience very different thermal environments. There are relatively fewer opportunities for that in the ocean. If we look at population abundance, apparently the most abundant vertebrate on land is the domesticated chicken, with something like 20 billion chickens around the world. Right? We like to eat chickens. But that's actually nothing compared to the 100 trillion organisms in some species of mesopelagic fishes. So, many orders of magnitude more. And that has consequences for evolution as well, right? Strength of natural selection relative to drift, relatively stronger, we think, in the ocean than on land. The final just qualitative comparison I wanted to make as we get started was to ask all of you a question as well. So who finds it easier to swim than to run? Can you just raise your hand? Yes. Okay, good. That's actually some more than sometimes when I've given this talk. I was at a marine station last week, and I think only 2 people raised their hand. I was astounded. Okay, who finds it easier to run than to swim? I fall in that latter camp. I was on the swim team in high school, and yet I'd much rather run than swim. For organisms that have evolved in these environments, though, it actually takes 8 times more energy to run an equivalent distance than it does to swim, and a lot of that has to do with buoyancy. So mobility in the ocean tends to be greater not only at the adult stage, like I'm showing you here, but also for even propagules. So larvae, the larval stage of many marine organisms float with ocean currents over kilometers, even hundreds of kilometers. So, I think this raises interesting questions about the impacts of global change on these ecosystems. What are the relevant, most important ecological and evolutionary processes, and how do these impacts compare to what we see on land? And so these are some of the questions that motivate the research in my lab. I'm trying to understand the ecology and evolution of global change in the sea. In today's talk, I want to focus on 4 questions in particular. First, from an individual organismal perspective, how does sensitivity to warming vary across terrestrial and marine species, ectotherms in particular? Second, do differences in sensitivity scale up to population level responses to variation, in particular variation in their thermal environment? Do those changes then affect biodiversity at the community level? And finally, I want to wrap up by thinking a little bit about the implications for conservation and for people. So on this first question, it's useful to think in terms of a thermal performance curve where there's a minimum temperature where an organism can survive, as well as a maximum temperature. And then we can define sensitivity to warming in terms of something that's been called the thermal safety margin, which is the difference between the body temperature of an organism and its upper thermal tolerance. So we can start out by looking at how upper thermal tolerance varies across species in marine and terrestrial organisms. And in collaboration with folks like Jennifer Sunday, we've pulled together data on upper thermal limits across ectotherms. About 320 species on land, about 90 species in the ocean, from reptiles to amphibians to insects, fish and crabs. And just plotting those upper thermal limits, so the y-axis across latitude, terrestrial organisms on the left with the lizard, marine organisms on the right with the blue fish. We can see that those upper thermal limits relatively flat across latitudes on land, and maybe dropping a bit more towards the poles in the ocean. We'll say that these measurements, these are collected from the literature, from lab experiments that have been done. There are some differences, important differences in lab methods that have been used across different species. We've tried to correct for some of those differences, including acclimation temperatures that have been used. The other important part of this, though, is the body temperatures that organisms experience. And one of the things we know is that it's actually not the average temperatures that matter most to organisms, but the extreme high body temperatures that they're likely to experience. Those extreme events have the greatest impacts, and actually are also most relevant to those acute lab thermal tolerances that get measured as well. So defining body temperatures and calculating body temperatures on land is a bit tricky, It depends not only on, it's not the same as air temperature, it depends on solar radiation, convection, conduction. So the calculations I'm going to show you are for, as well as behavior, and what habitats an organism chooses. So the calculations I'm going to show you are for microclimate models run for shady locations at the locations that each organism was collected from. We're looking at extreme high body temperatures calculated at an hourly scale. Models put together by Michael Kearney and Warren Porter. So I showed you the black dots and the black line before. I should say the dots are the individual data points for each organism. The line is a generalized additive mixed effects model. Now the orange dots are those extreme high body temperatures calculated in the shade. Again, line from a generalized additive model. And you can see there's maybe some wiggles across latitude. And it's actually interesting if we zoom in, though. So just zooming in on extreme high body temperatures for a second. The thick black line is the average fit across latitudes. And it looks like there are actually 2 peaks there. On the other hand, right, there's some uncertainty about the exact fit, especially when you're fitting a line like this. So each of those gray lines is a sample from the parameter uncertainty of that fit. So we can pull 1,000 fits, potential fits from the parameter uncertainty and count up. How many peaks do each of those have? Is it 1, 2, 3, or 4? It's a histogram here across the right. We actually see that there's actually pretty good support for 2 peaks, not 1. We can also plot a histogram of where those peaks occur. And what we see is they're not occurring at the equator. They're actually occurring at slightly higher latitudes, which to me at first was confusing. And then I realized that's actually where the hottest hours occur. Hot, long summer days heat up the most where they're longer, at slightly higher latitudes. We can then look at upper thermal limits. We also see 2 peaks not happening at the equator. Some indication that actually these upper thermal limits are also mirroring extreme high temperatures across latitudes. So this is terrestrial ectotherms. If we look at marine ectotherms, convection and conduction, very rapid processes in ocean. Body temperature of marine ectotherms matches water temperature quite closely. Those are easier to calculate, therefore. So then we can plot for terrestrial and marine ectotherms. We have the 2 components for calculating thermal safety margins. So it's just the difference between those upper thermal limits and extreme high body temperatures they're likely to experience. I will say that the marine values as well, we've tried to account for the potential for behavioral thermoregulation in the ocean based on mobility and the habitats that these organisms experience, though there is in general less known about behavioral thermoregulation in the ocean. So if we look at those thermal safety margins now calculated across latitudes, in green we have terrestrial thermal safety margins, and we see that the slimmest thermal safety margins are at these intermediate latitudes, about 20 to 30 degrees north and south. In the ocean, though, across all latitudes, they tend to be thinner than they are in terrestrial species. We can also think about how much those hottest hours are likely to warm going forward using projections from global climate models, and we see that also in the future. At least this is showing a high greenhouse gas emission scenario. We expect thinner thermal safety margins in the ocean than on land, though there's a bit faster warming predicted on land than in the ocean. So at least at this sort of organismal level, when we're accounting for physiological tolerance, behavior, and as well acclimation abilities, which differ slightly between marine and terrestrial ectotherms, it suggests that marine species are more sensitive to warming than are animals on land. The question then, though, is does that have population level consequences? Do we see those kinds of impacts mirrored at a population level? So if we think about a population spread across latitudes, some warm range edge and a cold range edge, and the set of processes by which species cope with environmental variation down here. So from behavior to physiological tolerance, acclimation, even evolution. It's those set of processes that help determine whether or not that warm edge stays in place as temperatures increase or whether it contracts to higher latitudes. So we might expect if at this organismal level marine species are more sensitive, then do we see faster rates of range contraction in the ocean than on land? To try to answer that question, we can look at data where species distributions have been surveyed through time, 2 times or multiple times. This is just an example from the Northeast US. So Rutgers University right here, sort of in central New Jersey. We've got North Carolina down here all the way up to Canada. It's about 1,200 kilometers across this span. These are from ecological surveys on the continental shelf. I'll talk about them a little bit more later on. But these areas in red are high densities of longhorn sculpin, a species found on the continental shelf. And we see that in the late 60s, the southern range limit was around Virginia. But by the end of 2017, that had contracted about 300 kilometers north up towards New York. So we have examples like this. Northeast US is interesting in part because it's been warming very rapidly over the last decade or so. But I've gone through the literature and found examples where we have resurveys of species warm range limits, both on land and in the ocean, focusing in particular on multi-species studies. to try to avoid or at least limit publication bias publication bias towards just publishing ranges that have contracted. We have data from about 160 species. From the marine species, about half of those ranges have contracted to higher latitudes. In the ocean, on land, about a quarter of them have contracted to higher latitudes. And we can try to account for some of the potential differences between these studies and between these species, including taxonomic similarity, the latitude at which these range edges were found early on in the study, the duration between the resurveys, whether they were resurveyed twice or more than 2 times. But after trying to account for those things, yes, it seems like this is a significant difference between marine and terrestrial realms at this scale. As I argued earlier, there is some evidence, though, that marine species are more mobile. So, is this just mobile adult marine organisms that are choosing new habitats? Is that why it seems like they're contracting more to higher latitudes? So we can also trim the marine species down just to the 60 or so species that have low mobility as adults. And yet we see actually equivalently high rates of range contraction, suggesting that this is not just adult movement, but actually extirpation, at least in many of these cases. So if we're seeing more frequent range contraction, warm range edges in the ocean, right? Those range contractions can actually cover the entire species distribution. And yet whether or not that leads to extinction depends on what happens at the other end, right? Whether colonization is making up at least some of what's happening at the warm range edge. So that brings us to the other set of processes by which species cope with changes in their environment through space and through time. So I want to address this question in particular by asking, do species distributions follow thermal habitats as they move across space? There's research that I've been doing for the last few years, that my lab has been doing, focused in particular on continental shelf ecosystems. So the light blue area, sort of that halo around the continent, the shallow parts of the ocean, where high diversity as well, high productivity, and also the parts of the ocean actually that have the strongest interactions with people. But one of the things we've been doing is collecting and compiling and standardizing ecological surveys from the last half century in these ecosystems. So each of the areas in red represents one of the surveys that we've pulled together. These are largely conducted by the National Marine Fisheries Service, at least in the US, Department of Fisheries and Oceans in Canada. These are bottom trawl surveys. So this figure is looking at the back deck of one of the research vessels. They drop a net off the back, pull it near the bottom, pull it up to the surface, and count and weigh the range of species that are found. And they sort them on deck. And it's everything from sort of familiar species like lobster and cod, as well as a very wide diversity of other species, whelks and sea anemones and all sorts of sea pens, all sorts of other things. It's actually an impressively good community survey, at least for the community near the bottom. So just to summarize a fair bit of analysis relatively succinctly, we can plot, again, we're back in the northeast US, but we can plot the center of a species distribution, in this case, black sea bass, as a dot on this map. So this is where the center of black sea bass distribution was in the late 60s, and then this arrow points to where it ended up by 2008. In this case, black sea bass were shifting to the northeast at about 50 kilometers per decade over those 40 years. But as I mentioned, these aren't, this survey is not focused on one species, but actually the diversity of species that are on the continental shelf. So each of these orange arrows here, again, the circle is where the center was in 1968, the head of the arrow is where it was in 2008. And you can see there's a lot of diversity across species in terms of the rate and direction that these species were shifting. Some species like black sea bass shifting fairly rapidly to the northeast, other species not moving much at all, still others even moving south. We've got squid right in here shifting to the southwest. We can also zoom out to sort of the continent scale, and again, we see a lot of variation. In some regions, like the northeast US, on average, the assemblage has been shifting north over the period of these surveys, but in actually a larger number of regions they've been shifting south. And this is actually fairly similar to a range of similar data sets for small mammals in California or plants across North America, where there's a lot of variation from one species to the next in the rate and direction in which their distributions have been changing over time. And a lot of that has been attributed to basically idiosyncratic shifts in species distribution, right? It depends a lot on the exact physiological tolerances of the species involved, their interactions with other species, maybe lag effects as well. But I also was curious whether there may be some of this variation that is actually predictable, in part from this idea, this concept that's called climate velocity. So climate velocity is the idea that the world is filled with temperature gradients. So there's a warm edge of a gradient, a cold edge. If you pick a particular temperature, you could draw that isotherm on a map. And then as that gradient warms or cools, that isotherm is going to move at a particular rate and in a particular direction. And the combination of rate and direction is what we call climate velocity. So we can basically calculate from temperature records climate velocities in each of those locations, and then compare them against the rate at which species have been shifting. So the vertical axis here shows the degrees latitude per year that species in each of these regions were shifting, either north, south, or not moving much at all. And then the horizontal axis is climate velocity, either pointing north, south, or not changing much at all right there in the middle. Each of the gray dots is one of the taxon within one of the regions around North America. And we can see there's quite a bit of variation across species as well as across regions. The red line, though, is not a best fit line. It's actually a one-to-one line. So if species followed climate velocity perfectly, they'd be right on that line. Clearly, they don't, right? There's a lot of variation, a lot of things that drive shifts in species distribution, colonization rates, extirpation rates, beyond just changes in temperature. And yet, we can explain about 40% of this variation across species just based on this one factor, climate velocity, suggesting that at least a decent amount of this heterogeneity across species and across regions can be explained by the thermal landscape that these populations inhabit. And accounting for that variation in thermal landscape is a really important part of understanding what's going on here. So this is across latitude. I can show you the same thing, but across depth as well. So just as on land, terrestrial species are moving towards higher elevations in many cases in the ocean, when surface temperatures warm, many species are moving deeper. And we have some ability to explain variation across species in terms of their movements across depth based on climate velocity as well, though relatively less than across latitude. These same data also allow us to ask another question. Do populations in the ocean lag behind climate velocities? Or are they alternatively keeping up? So if you go back to this graph for latitude, species in the lower right quadrant are not shifting north as fast as we'd predict from climate velocity. And the degree to which they're lagging is the difference from this one-to-one line. Species in the upper left are not shifting south as fast as we'd predict, and then species in these other two quadrants are actually speeding ahead of what we'd predict just from climate velocity. So showing a histogram of those differences, either speeding ahead or lagging behind. On average in marine species, we don't see evidence for either, speeding ahead or lagging behind. And we've spent a fair bit of time trying to understand characteristics that may cause species to end up on one side of this histogram or the other. And the most important factor we found is the growth rate of the species involved. Fast-growing species less likely to lag behind. So, actually what you might expect from invasion biology. Yet I have to say the effect is really, really small. So I think the more interesting contrast is actually that we're not on average seeing strong lags in the ocean. Whereas on land, from a range of studies, there is evidence that many terrestrial species are lagging behind climate velocities. Birds to butterflies, plants, trees, and small mammals. And that may have to do in part with greater mobility of marine species, ability to colonize new habitats, as well as what I showed you earlier, sort of greater rates of extirpation at warm range edges, or either range edge, depending on which direction temperature is changing. So what I've been showing you is decade scale or multi-decadal scale shifts in species distributions, relatively long-term patterns. So it also got us interested in whether species are responding very quickly to variation in temperature as well. Marine species are, very much exposed to changes in temperature of their environment. So to get at that question, we looked at similar data sets to what I showed you earlier, but instead of looking at long-term trends, we looked at the deviations from those long-term trends. So this is just one example. Atlantic moonfish is a fish found in the southeast US. And the vertical axis, the y-axis here is standardized biomass in this case, just within the region in a given year. And so deviations in a given year are just the red lines here. So relative to the long-term trend, are there relatively more moonfish or less moonfish in a given year? And then we can compare those to the temperature of their environment. And we've looked at these particular surveys happen in the spring and the summer. And we've looked at temperature in different seasons, and it's winter temperature that's actually most important. It explains the most variation, not only for moonfish, but actually for other species as well. It seems like winter temperature ends up being that limiting, the limiting season. But we see, at least for moonfish, there are more moonfish after a warm winter, fewer after a cold winter. So we're interested, though, not in just one example, but across the assemblage. So we can plot that positive response to winter temperatures on this vertical axis. Positive response to winter temperatures for moonfish. Shrimp also have a positive response as well, but other species like smooth dogfish have a negative response. Fewer dogfish after a warm winter. And across that assemblage, again, there's a lot of variation, right? What you might expect. It's interesting in part because we hadn't quantified that kind of response to winter temperatures across an assemblage. But what's more interesting then is trying to compare that to, trying to understand that variation across species. And what we see is that using independent data on species thermal preferences, we see that species that prefer warm temperatures are showing positive responses to a warm winter. Species that prefer cold temperatures are showing a negative response to warm winters. And then species in the middle are showing not much response at all. That shows up statistically as an interaction. So in a mixed effects model, it's the interaction between the winter temperature and the temperature preference of the species involved that helps explain that variation, actually at an interannual level. But I think what's important to recognize, though, is that the mechanism really varies a lot between species. So for some species, it appears to be overwinter survival that is creating this response. So like for star drum, they overwinter in shallow estuaries. Few of them survive after a cold winter. So that cohort after a cold winter is smaller out on the continental shelf. More of them after a warm winter. Whereas for something like smooth dogfish, it has to do with migration phenology. So after a warm winter, they're starting their migration earlier and are actually out of, or partially out of the southeast US by the time this survey comes through in the spring and the summer. The migration is happening later after a cold winter, and more of them show up in the survey. So very different mechanisms, ecological mechanisms, even if it's a relatively general pattern across species. So I think that the take-home message is that we're finding evidence that marine populations are responding very rapidly to temperature variation, especially through these processes of colonization and extirpation. And that when we can make the comparison, they seem to be doing so faster than what we see happening on land. And the question is, how do these changes at a species level scale up to affect community diversity? And I think that there's actually been quite a bit of debate in the ecological field about how diversity is changing through time. We hear a lot about declines in diversity and extinctions, and yet we also have plenty of examples where actually diversity in a community is going up. For example, on islands, humans bringing new species to islands has driven up diversity. And yet a lot of that research is happening either at a very local scale or at a global scale. There's been relatively less done at a regional scale. So we were curious, how is diversity changing in these 9 regions around North America, in these continental shelf ecoregions, or ecosystems? So we started out by quantifying just species richness. And yet, obviously, species richness is actually quite challenging to calculate, in part because detectability of species varies, some of which are not necessarily well detected by the bottom trawl surveys that we're using for these calculations. So what I'm showing you here are output from a multi-species occupancy model to try to account for those differences in detection across species. If we look at the Northeast US, we see that richness has been going up over the last few decades. And actually, across 8 of these 9 regions, we see that richness actually has been going up through time. Those trends are statistically significant in 4 of those regions, but all 4 of those are increasing. None of them are decreasing. So I think that's really interesting. It's in part because it's driven not by humans transporting new species into these regions, but largely by the greater persistence of species that are rare. Species that used to blink in and out early on in these data sets are now present more frequently, and therefore are contributing to species richness in a given year. We can also look at where these colonizations are happening. So this map, again, for the Northeast US, in red shows where most of the colonizations in this region have been happening. In this case, it's at the southern edge of the region, right at Cape Hatteras, which is also a biogeographic break. So what you might expect, as new species move into the region, that's where they're colonizing from adjacent regions. In other regions, we see colonizations also happening at the edge, but not always, I should say, the southern edge. Sometimes it's actually at the northern edge, species coming in. Tends to be associated with where there are biogeographic breaks and sort of other species with range edges near the edge of the survey region. What's also interesting, though, is that when we compare the rate at which richness is changing, so that's the y-axis, against the rate at which temperature is changing in each of these regions, we see a positive association. And it doesn't actually depend on the direction in which temperature is changing. So this is just absolute value of temperature change. More rapid increase in species richness in places where temperature is changing faster. And at least my interpretation of that right now is that we're seeing transient dynamics. We're seeing after temperature changes, we're seeing rapid colonization by new species moving in, but slower extirpation of the resident species. We're still trying to do work to follow up on this to see if that hypothesis holds. But I think these may be transient increases driven by changes in temperature. New ecological opportunities opened up, but species that are there haven't yet disappeared. The other thing that process implies, though, that there's changing composition of species. So if temperature goes up, we expect warm water species to be moving in and cold water species to be moving out. And one way to quantify that is something called a community temperature index. It's been used a few different times, especially for birds and butterflies, but other ecosystems as well. And the idea is that sort of you look at the thermal preferences of the species in a given location at a given time, sum them up, divide by the number of species. So it's sort of an average thermal preference for species in a location at a particular time. So just to go back to the Northeast US, we've quantified how the community temperature index has changed through time for different parts of the Northeast US. In purple, there's been a shift towards species that prefer cooler temperatures. In red, there's been a shift towards species that prefer warmer temperatures. Again, a lot of heterogeneity across even different parts of the Northeast US. We just zoom in to show you an example. Water temperatures in that particular location have been going up through time, and the community has shifted more towards species that prefer warmer temperatures in that particular place. On average, though, we see about a third of a degree change in the community temperature index, so community composition, for every 1 degree change in water temperature, which I think actually is an impressive amount an impressively large response given how many other factors influence these ecosystems. Food web dynamics, predator-prey interactions are incredibly important in these dynamics, in these ecosystems, as well as just stochastic recruitment, and for some species, intense impacts from fishing as well. Even more interesting, though, is I think the responses, how the rate of response depends on community composition. So if we think about the community temperature index on the y-axis, either greater proportion of warm water species on top, greater proportion of cold water species on the bottom, across water temperatures on the x-axis. If community composition followed water temperatures perfectly, it'd be right on this 1-to-1 line. If there was no response, it would just be a flat line, or just a cloud. And yet what we see is actually this nonlinear response across temperatures, sort of a less than 1-to-1 response at low temperatures, greater than 1-to-1 in the middle, and then less again at higher temperatures. And I think why that's happening actually has to do with the species in this sort of the regional species pool, which is made up in some ways of two guilds of species. There's a histogram of species temperature preferences across the top. So we've got a guild that prefers cooler temperatures down here, a guild that prefers warmer temperatures up here. People have seen similar multimodality, even in global data sets of marine species temperature preferences. And yet we see, where we see these rapid turnover in community composition is where we see this turnover from the cool water guild to the warm water guild. And slower responses when we're fully within one guild or the other. So I think the take-home message is at this community level, we're seeing actually species richness is increasing, especially where temperature is changing, whether warming or cooling. And in some cases, we actually even have some predictability about the community composition and how it's changing. So I just wanted to end by talking a bit about implications for conservation and for people. So I've been arguing that and sort of showing you data that in many cases marine species are on the move towards higher latitudes as temperatures warm. And this map here shows in red how much further we expect marine species distributions to shift by the end of this century, at least under a high greenhouse gas emission scenario. And at least in the ocean, most marine conservation is not actually accounting for these dramatic changes in where species are found or in the composition of local communities. For an average protected area in North America, we expect about a third of those species to be gone that are there now, to be gone by the end of the century, replaced by a more or less equivalent number of species by the end of the century. That also means, though, that setting up networks of protected areas across gradients of latitude and depth provide an opportunity for species to, in effect, hop from one protected area to the next along sort of as stepping stones as those distributions shift. Even more, though, than necessarily these sort of static conservation approaches, though, is I think it's very important to recognize that, especially in the coastal ocean, not only are these ecosystems strongly impacted by human behavior, but that feedback actually goes in both directions. That human behavior is also strongly influenced by the state, by the dynamics of these ecosystems. These are strongly coupled social ecological systems. So that's, in my own work, that's led me to collaborations with social scientists, which has been really interesting in many cases. So collaborating with geographers and with economists and with anthropologists to understand how both sides of this coupled system are working together and changing through time. And just to show you one example, large fishing boats from Beaufort, North Carolina, right here, in the mid-90s, on average, were fishing off North Carolina, where this red square is. By 2014, though, those boats were steaming on average 1,000 kilometers round trip in search of summer flounder that were relatively rare off North Carolina, but have become quite abundant off New Jersey and further north. The shift in species distribution appears to be partly because of warming water, but also has to do with other dynamics, including recovery from historical overfishing. It's not just climate that's driven that northward shift. But it has been a very dramatic response in fishing behavior in these communities, in these fishing communities. And that shift in species distribution actually has driven increasing conflict between states arguing over who should have access to this species. Should it be the fishers who had access to it historically, or should it be the fishers who are now close to where the species is most abundant? But and we see similar conflicts now playing out even internationally as well. So I think one of the more striking examples has to do with Atlantic mackerel. It's a species that historically has been fished by Norway, European Union, and Faroe Islands, but then shifted north into Icelandic waters. And Iceland and the European Union couldn't agree over how to share access to that fishery. They both fished how much they wanted, led to overfishing, led to declining abundance. In addition, what started in fisheries didn't stay in fisheries. It spilled over. It became a trade war. It also helped convince, it appears to have helped Iceland that didn't want to even join the European Union at all. They initially had a bid to join the European Union. And they're actually still, that started about a decade ago, and they're still actually not cooperating. And I think that sort of illustrates this broader point that as species distributions in the ocean are shifting, when that influences fisheries, those fisheries movements across political boundaries are in many cases leading to conflict. So that got me wondering, how much do we think this might be a problem in the future? So we can take species distribution models, couple those to projections of future environmental conditions, or climate models for the ocean. And then each of the red areas here is an exclusive economic zone, so sort of a nation's waters, projected to have at least one new transboundary species by the end of the century as a result of climate change. We can see that they're spread all over the world. Shown a different way, the number of exclusive economic zones with a new transboundary species against the rise in global atmospheric temperatures, either high greenhouse gas emissions in red or lower in blue. We see that the fewer emissions are put into the atmosphere, the less potential for conflict there is going forward as well. Even beyond lower greenhouse gas emissions, though, we also know there are other approaches that can help avoid conflict. From ecology, evolution, and climate science, we can help understand where there are likely to be these problems and conflicts break out over shared common pool resources. Study of institutions help us understand how to build trust, including by sharing data and sharing science. And study of human behavior, game theory, also help us understand how to structure those negotiations effectively. There aren't clear, easy solutions, but there are actually some good examples around the world where countries share transboundary species relatively well, including tuna in the Western Pacific and Norway and Russia in the Barents Sea. I think more generally, the next few decades are going to be a key time for determining what happens to marine ecosystems, both from perspective of climate variability and climate change, but also from the perspective of human behavior. And I think one of the challenges is, can we bring science to bear on these questions to help society make informed choices going forward? I think that's a really important thing and a really important problem for people to tackle. And I just wanted to end by talking a bit about sort of some of the key areas where I think research is needed to help answer some of the questions. I think those questions. One key area I think has to do with understanding not just how single species are responding to climate variation and climate change, but how entire communities are changing. Why are some communities turning over faster than others? Are transient dynamics a key component of the changes we see over the scale of decades? What role do they play? What does that mean for the long-term trajectories of these species? And I've shown you some initial results today, but I do think these long-term community data sets we have for continental shelf ecosystems are a really valuable resource for digging into these questions. Showing you results from North America, but they're actually similar data sets that we're now starting to look at in Europe as well, so we can look across different continents as well as different oceans, and trying to understand what are the predictable components of community responses, despite very different species compositions. The other key question, I think, is, and you'll notice, in the very beginning I presented evolution as an important way, potentially by which species are responding to changes in their environment. Marine species, because they have high abundance, because they have lots of gene flow across environmental gradients, there's good reason to think they have lots of standing genetic variation that may allow rapid evolution. But we actually don't know very much about whether that's happening, what role that plays, and the ways in which populations are coping with variation in their environment. So the approach I've been taking or my lab has been taking, is to look actually at historical samples, where we can look at DNA sequencing and then look at allele frequency changes across the genome through time, to try to observe, in effect, rewind time and look at evolution as it happens in response to environmental change. So obviously a lot of challenges there, connecting genomic change to phenotype and fitness and all of that, but I think really fascinating challenges as well. Doing some work with Atlantic cod, where they now mature at younger ages and smaller sizes, presumably as a result of fishing. Also looking at bats that have been recently infected with a fungus. Populations have declined rapidly, but there's some evidence that they may be recovering. And then finally, just starting a project looking at coral reef fishes in the Philippines, where they've been strongly impacted both by habitat change and fishing. But there's an opportunity to compare across a number of different species, all from the same location. The final component that I think is really important is understanding persistence at a much more local level, right? So demography, metapopulation dynamics are the processes that underlie both shifts in species distributions and also changes in allele frequencies through time. So reproduction, dispersal, survival. And yet in the ocean, one of the fascinating things is that most populations release their offspring up into the water column and then they float away. There's some small fraction that comes back that's retained, but we don't actually know whether that's enough to ensure self-persistence, or whether actually persistence is a metapopulation level phenomenon in these cases. So my lab has been studying anemone fish in the Philippines for the last few years, trying to understand both dispersal, so something we do with genetic parentage analysis, as well as reproduction, growth, and survival we can do with tagging and with field work. So just to summarize, or what I've been showing you today, showed you with a synthesis of physiological data at an organismal level, that it's suggesting that marine species are more sensitive to warming than our terrestrial species. Then, using long-term ecological records, showed you data suggesting that marine species are responding very rapidly to variation in temperature, especially through colonization and extirpation dynamics, and that those seem to be happening faster than what we see on land. At the community level, that we're seeing actually increasing species richness, especially in places where temperature is changing rapidly, and that there's some predictability in community composition as well. And then finally, that actually in the ocean especially, we need to apply new approaches to conservation, especially those that account for the feedbacks between people and the environmental changes, and thinking about those coupled social ecological dynamics. So with that, I guess I just wanted to wrap up by pointing out that both on land and in the ocean, right, it's the same ecological and evolutionary processes, but they're very different environments. And in many cases, I think those help to explain the very different outcomes in some cases that we're seeing. And I think more broadly, comparing and contrasting across realms is a really rich source of insight into dynamics and processes in both ecosystems. Just wanted to thank a wonderful set of collaborators on the research I've shown you today, the folks in my lab who've done a lot of the heavy lifting that I've shown you and make all of this fun. Especially, I showed you work from Ryan Batt, who's a postdoc in the lab, Rebecca Selden, who's currently a postdoc in the lab, headed off to Wellesley College in July, Jim Morley, and Patrick Flanagan. And then finally, just to thank all the funders who make this possible. Thank you.
Questions? Yes.
Well, you seem to have concentrated on macrofauna.
Right.
Yeah.
Does this work?
Yes.
Yes.
And so you occasionally mention plants. You did not mention microbes in the context
Good point.
of
Right.
this. And one of the things that, well, there are two things that concern me about it.
Okay.
When you reckon biodiversity, do you include plants in the biodiversity, or are you just talking about the macrofauna?
Right. So when I was talking about species richness, that was specifically for the animals that are caught in the bottom trawl surveys. It doesn't include plants and does not include microbes. Yep.
All right. Further, when plants move out of an area and there are animals that depend on those plants,
Right.
then the change in temperature that presumably has caused the plants to move has only indirectly affected the migration of the animals.
Mm-hmm.
So how are your conclusions
Yeah.
affected by that?
So, right, are the patterns we're seeing, are they direct effects of temperature on the organisms, or are they indirect effects? Is that sort of a way to rephrase
Yes.
what your question is? Right. Well, on one level, we don't necessarily know. right? We've made the connection between the rates and direction that temperatures are changing and the rates and directions that species distributions are moving. But it doesn't necessarily tell us the mechanism. I do know, though, I mean, based on what we understand about the system,
Yes.
I think it's unlikely, though, that they're just following their prey. So most of the species we look at are quite generalist predators. They're eating a wide range of taxa. And actually, their diets, in some cases, that we're just starting to look at are changing as well, based on what's available, not necessarily that they are moving to follow their diets. I think cases where, though, in the ocean where that does start to become an important process are especially when we look at seabirds. It's not research I've done, but other folks have done some really interesting work with terns and with puffins, especially in the Gulf of Maine. So when their chicks are young, parents are bringing small fish back to feed their chicks. And when butterfish move in, it's a southern species. It's relatively, basically the body size is taller and it doesn't fit down the gullet of a chick very well. So there it's an indirect effect. Basically, different species composition, they can't feed their chicks well, and they see basically a year class that has very low fledging success. Yeah.
Okay, I guess I have it.
You do.
So that was really interesting. Thank you. I'm really curious about the conclusion that there are differences in rates of the marine animals seem to be tracking climate more than. Can you sort out the difference between you used a single point to show them moving,
Right.
but of
Yes.
course there are the leading edge and
Totally.
the lagging edge, and the ideas you're talking about for colonization happening faster than extinction
Right.
can be looked at by thinking about that leading lagging edge. And
Yes.
part of why I say that in plants, at least along elevational gradients, we know that the colonization is happening a lot faster than the extinction. And the data are pretty good for that, and that's, as you suggested for these animals, that is the cause of observed increases in richness. So have you been able to sort out those two edges as you look at the range shifts?
Yeah. So actually something we're working on right now. So we haven't done it. The reason we focused on centroids initially was just because some of the range edge dynamics for some of the species are driven by changes in abundance. Many marine species have density-dependent habitat selection, so you see range edge movements just from changes in abundance, whether fishing or species interactions or other influences. So it's made it a little bit tricky, but I agree. And actually, I think it's, when we think about shifts in species distributions, it's, the paper I showed you was, 6 years ago now. It's more useful to think about what's happening at each range edge separately, right? Because we have different processes operating at each range edge. So, yeah, I totally agree. Nice throw. Nice catch.
Those 4 years on the bench in football in high school paid off. I'd like to echo the thanks for the interesting talk. And I was curious, do you see any effect of diet breadth on the rate of range shift? You might think that a specialist has an imperative to move if its food source is moving, or a generalist has the kind of like free diet space to look for new things to eat if it goes somewhere completely new, or can follow one set of its diet or a different set of its diet, and it might expand even more.
Right. Yeah, exactly. I think interesting questions about sort of invasion resistance or predator release also sort of going on during these shifting ranges. In the continental shelf ecosystems we've been looking at, there's not great contrast in diet breadth, at least in a way that seems to make a difference. There are good diet studies, though the data are a mess. So we've been thinking about going that direction, sort of quantifying diet more precisely. Folks like Jennifer Sunday have done a little bit of work in Australia suggesting that omnivores are colonizing new territory faster than either herbivores or predators. But yeah, I think there's a lot of work to be done to connect, especially across different ecosystems, diet breadth and other measures of specialization or generality to the rate of colonization. We think it should matter.
Sure.
Yeah.
Thanks.
Yeah.
Sure. never a quarterback.
Nice.
That's why I got a postdoc.
Oh, sure.
Great. Many of these populations, of course, have moved a lot in the past 20,000 years. And there's a pretty comprehensive marine fossil record for some of them, although I realize the sea level was somewhat different on the nearshore stuff. Have you talked to your paleontological colleagues about this? Because, I mean, you're talking about 5-degree change over 20,000 years, which is very robust. But the one change at the moment, though, is it's not just the temperature that's changing, it's also the pH.
Mm-hmm. And oxygen.
And oxygen.
Yeah, oxygen especially is likely an important part of that. I didn't mention it, but a lot of the temperature, the physiological effects of temperature on marine ectotherms probably is mediated by oxygen demand and availability.
Right, so I'm just wondering whether those particular drivers, especially with pH and perhaps oxygen, would give you a qualitatively different result from the post-glacial maximum rebound.
Well, so I can say that I've been working quite a bit with John Payne at Stanford, thinking about some of the mass extinctions over the paleo record and how those may be biased towards high or low latitudes. And there is some evidence that basically during mass extinction events characterized by rapid warming, like the end Permian, the species that were most likely to go extinct are those that were already at high latitudes. So Curtis Deutsch, actually from University of Washington, just had a nice paper along with John suggesting that that's driven by low oxygen at high latitudes. And basically low latitude species moving to higher latitudes where their habitat was, and the high latitude species running out of available habitat. And then actually there's some evidence, like during the end Ordovician, that the opposite happened. So about 5 degrees of cooling, and it was the low latitude species that were most likely to go extinct in the ocean. So again, at least suggesting in some senses that they were moving towards lower latitudes except for those that had nowhere left to go. So I think that's an interesting question sort of at that macro scale.
I'm not going to do that.
So you mentioned that there's something about the timing of the surveys. They made it so that you sometimes miss the dogfish because they migrate and stuff like that.
In that part, exactly.
How else does I was curious about how the timing of the surveys is relative to the breeding season of the fish, and how else that could affect your perspectives on the response of migratory fishes, especially if you think of migration as a form of escaping an environment for persistence.
Yeah, exactly. I mean, I think there are some interesting parallels, especially for some of these migratory species, between what's happening in birds and what happens in marine species. To what extent are they moving long distances as adults, but are they still tied to particular habitats for breeding and for feeding? I don't think that's actually very well understood in the ocean. Some species seem to do it mostly based on environmental cues, but others seem to be cueing off topography or particular bathymetric features that they're using to navigate, even if the timing may depend more on environmental cues. So, in the data set I showed you, most of the species are not migratory. Smooth dogfish are one of the ones that is, quite a bit. Black sea bass actually are as well, seasonally moving. And the surveys, depending on the species, line up or don't with breeding periods. So for the black sea bass and summer flounder, another species that's there, they're primarily near the coast and farther north in the summer. Farther south in the summer. Anyway, they're close to shore. And then moving seasonally. So, that interaction between the seasonal migrations and the timing of the survey is actually an important dynamic here as well. I don't know if that answers your question, but I think it's an important dynamic. Yeah.
One more? Last question.
Somebody else should get it. Thank you, Crystal. That was very kind of you. I was not a quarterback, nor was I on the bench, right? Okay. Just one thing you haven't talked about that is one of the other mechanisms that might be going on as the communities change is that you talked about each species kind of moving independently, but of course they're moving at different rates.
Totally.
And therefore the interactions are going to change. And one
Yes.
of the things that people have started talking about and doing experiments on is as you get these novel assemblages, how much are these changed interactions going to cause extinctions that might be independent of individual response to climate. And so that's going to really change our ability to predict how these assemblages are going to change. And what's your thought about how important that's likely to be? How do we go about looking at it?
So we already have some evidence that species interactions are changing. So just in the northeast US, for example, the mix of species that are important prey and